Hey, my name is Chase Curtis, and I'm going to be explaining the process between a neural stimulation and a muscle contraction. So first we have our neuron right here. This neuron is going to be stimulated by either the spinal cord or the brain, which is going to run to the spinal cord into the neuron. This neuron stimulated, and it's going to cause an action potential. So it's going to be excited, and it's going to open up sodium channels, and these sodium channels are going to open up, and it's going to become depolarized. If this depolarization reaches the threshold of a negative 55 millivolts, then an action potential will occur. When this happens, it becomes completely depolarized, goes up to positive 30 millivolts, and the action potential occurs. So the action potential is going to move down the axon, jumping between the myelin sheaths from each node of Ranvier. So 1, 2, 3, 4, moves all the way down to the axon terminal. In this axon terminal, you're going to have these little synaptic bulbs. So at the synaptic bulb, you're going to have the synapse, and you're going to have the motor end plate, which is the part of the plasmalemma of the muscle fiber. What's going to happen then is you're going to have a neurotransmitter that is released from the synapse, and it's going to bind to neurotransmitter receptors on the motor end plate. When this happens, it's going to also excite the muscle as well, and going to cause an action potential there as well. The muscle is going to become depolarized. It's going to open up ligand-gated channels on the motor end plate that are going to allow sodium to enter the cell. If enough sodium enters, then it's going to become depolarized, and it's going to reach the threshold of the muscle cell as well. If it reaches threshold, the action potential occurs, and voltage-gated sodium channels open up. So sodium is going to rush into the muscle cell. It's going to run down the T-tubules, and when that happens, it'll prime the sarcoplasmic reticulum to release calcium. So the sarcoplasmic reticulum is yellow right here. It's going to release calcium into the myofibrils. Myofibrils are full of two different myofilaments, which are smaller units of protein. So you have actin and you have myosin. The actin is going to be covered in troponin and tropomyosin. The tropomyosin is blocking the myosin from binding to the actin. So calcium is going to bind to troponin. Troponin is going to move tropomyosin, and it's going to allow myosin to bind with actin. So when this happens, you have what's called the sliding filament theory. So the myosin is going to bind to actin. ATP is going to bind to myosin, causing them to split. The ATP is going to hydrolyze, meaning that's going to lose it's going to be hydrolyzed, so it's going to lose a phosphorus group, become ADP and inorganic phosphate. And that's going to release energy. That energy release is going to cock the myosin head, move it back, change its conformation, and then it's going to lose the inorganic phosphate. It's going to bind to the actin again, and it's going to lose the ADP, and the head's going to change back, and it's going to move the myosin in a movement called the power stroke. So it's going to move the actin filament closer to the M line, which I'll show you right back here. M line's right here. It's going to move it closer, shortening the muscle, contracting the muscle. And when this happens, you're going to have another ATP molecule. It's going to bind to the myosin and separate it again. Same process goes on and on and on, as long as they're still stimulated, as long as there's still calcium inside of the myofibril. Now, when this process is done, there's no more neural stimulation. The calcium is going to be pumped back into the SR. Now, this process is an active process, meaning it needs ATP to occur. So the ATP is going to be used to actively pump calcium back into the SR. And when this happens, the troponin is going to lose the calcium. They're going to unbind. Tropomyosin is going to move back over actin. Myosin is not going to be able to bind, and the muscle is going to relax.